Hey, good morning, everybody. Thanks for being here. This is another seminar in quite a series that we have been organizing, and actually I want to take the opportunity to thank Isabel for following up on the seminars. If you have any idea for your own to present or any speaker you think should come and give a seminar, you can always float the idea. Today we invited Luc Dendove. Luc Dendove is a very esteemed colleague with whom we have been working over the past years in a very fruitful collaboration between CIMMYT and Simbestaff. When you see some of the numbers about publications coming out of the long-term trial infrastructure, the long-term trials we have here in Mexico, part of those publications come from student collaborations that we have been doing together with Simbestaff and Luc's research team. What I always say is the uniqueness is Luc has a lot of nice ideas, nice research tools that we don't have, and we have the long-term trials that can actually be the background or can generate a story through using those insights that are way more upstream than what we are doing. Luc has a very long and recognized career. He started its work at the University of Leuven, then was quite some time active in Rothamsted, and finally landed in Mexico in Simbestaff, where he's still leading one of the most important research teams on carbon-nitrogen cycling and recently microorganism processes, but I'm sure he will explain that way better than I can do. So thank you, and let's give a round of applause.
Good morning, and thanks for coming. I try. But okay, that's the title of my talk. I mention that on the bottom because if you read some of the earlier papers, then people try to give certain characteristics to bacteria. And I try to show you that, for instance, they are copiotrophic, they are oligotrophic. I'm not so sure about that anymore. I think bacteria are much more flexible. I don't know if soil characteristics are important in that. It might be competition. It might be predators. I'm not sure about it. So I try to explain a bit to you. So this is a paper that is ready to submit. It's a collaboration between different institutes, some younger researchers working in Tlaxcala and working in Polytechnico. Brown mentioned it already. It's rather a good collaboration. I'm quite proud of it. We have now 31 published peer-reviewed manuscripts. We got 745 citations. Some of these citations come from some very important papers Bram wrote. We have an index of 14. So I'm quite pleased with this collaboration. We have quite a lot of other papers ready to submit. Okay, we know that agriculture practices affect yields and soil characteristics. We studied some of these in El Batan and Obregon, but I focus mostly on El Batan. There's one pitfall in El Batan. Of changes in soil characteristics. We have one problem in El Batan, I mentioned it immediately. We got two replicated plots. So if you do an analysis, a statistical analysis, then you get problems because the variation is rather Great. So often you see clear-cut effects, but they are not significant. Yeah, some of the problems. That's one of the commands of the last paper that we submit. Doesn't even know it is accepted, but this is a command I often receive. So I try to explain that to the reviewer. Well, we can do statistical analysis, and we know, but I'm a bit worried about it because it returns and returns. That's why we try to work in Obregon. The problem with Obregon is that if we want to get some soil samples and extract DNA, then it becomes difficult. And if we want to extract RNA, then it's even more. Okay. We were expecting that these differences, especially in organic matter, would have a large effect on carbon and nitrogen mineralization. This is a very simple representation of the aerobic incubation we do. We got some soil, we trap carbon dioxide, and we measure in the soil mineral N. And now recently we start to extract the DNA to see what is happening there. Another command that I was not really aware of is that if you start to work with bacteria, then you have non-parametric data. I've never, did a lot of research on soil characteristics, and parametric, but bacteria, if you count, they're non-parametric. And again, if you try to do some analysis, then, significant difference is rather difficult to obtain because non-parametric or more stringent, if I can say it like that. Okay, so I said I would mention the problems we are encountering. These are some results of carbon and nitrogen mineralization. You see not a lot of differences between your treatments, although you have a difference in carbon content. And I compare that with Broadbork and Rothamsted, where after, how long was it? More than 160 years, you really have a big difference in carbon mineralization and nitrogen mineralization because your carbon content is extremely different. I just want to mention, you know that David Jenkinson published a paper on microbiomass carbon. There's no replicate in Broadbork. So if you look at the data on his original paper, then there's no replicates there. Additionally, he includes one soil with a huge amount of carbon. And you look at the line. So, but anyway, I think it was a very important paper, but I have some questions. Anyway, we didn't detect a big difference in carbon nitrogen mineralization. We did some measurements also in the field. We did that quite a while ago, and that was one of the problems we faced when submitting these papers on carbon dioxide emissions, and nitrous oxide emissions, and methane emissions, that review was pointing out you only have two replicates. Okay, this is the nitrogen we measured in the field. We don't have a lot of difference there. As we detected in the lab, so we think we need some more time to see an effect of conservation agriculture versus conventional, to see an effect on carbon mineralization and nitrogen mineralization. But okay, you have these data, you have these differences. So what is the effect of these differences on microbial populations? And this is a principal component analysis. And it's you try to get all your data on bacterial groups. You try to correlate them, and you place your treatments that you have in this field, how do you say that? In this area, or whatever. So what you get is you get a clear separation between permanent beds and between conventional beds. And what do you get in conventional beds? You get copiotrophs, which are organisms that like fresh organic material. So they consume it, they degrade it, they mineralize it. And if you go into permanent beds, you've got these organisms that they prefer nutrient depleted environments. With organic material, I have to point out that oligotrophs might be questionable as a word, because normally it has more to do with nutrients than with organic material. But I use it here also for organic material. So if there's a microbiologist, I apologize for using that word because some people are very sensitive to these words. Okay, so some people working in microbiology and bacteria, then you get vermicultus, which is bacillus. They're rather easy to isolate. If you read papers on isolation of microorganisms from soil, you will mostly find these, not few the same, mostly these. Acidobacteria is a huge group of microorganisms, but we only have isolated about 20. Why is that? These organisms are very difficult to grow on agar or in a flask. So it's clear that the agriculture practices do affect our microbial populations. As I mentioned, permanent beds, they favor these groups, which are oligotrophs that prefer, I think this is the opposite. They prefer nutrient-rich, this should be reversed, but anyway. And then we have conventional beds where we get these organisms that prefer organic rich environments. Why? Because if you have conventional beds, then you incorporate the organic material, so microorganisms get in touch with the organic material. If you have permanent beds, then you leave the organic material on top. There's no direct contact between your microorganisms, or very slow contact, or very low contact. Okay. We did a lot of experiments, so I mentioned. So I tried to compare a sampling we did in 2014 and a sampling we did in 2012, and it still works. So if you compare sampling in 2012, 2014, we'll get the same results. Permanent beds preferred by oligotrophs, they are favored. And if you go to conventional practices, I always have questions, is this for real? Do you have changes over time? Are my results correct? Because you always have this problem of technique. And what do I mean by technique? When we did these first analyses, we had clones. We had about 150 sequences of DNA. And then we went to Piro, which is about 100,000 sequences. And now we go to Illumina, and then we can talk about whatever you want, 400 million, 200 million. So the question is always, there's an enormous evolution in this field. It's tremendous. It's quite exciting, it's interesting, because we're fed up with carbon and nitrogen mineralization. But then you always have the problem, can I compare really my results that I obtained a couple of years ago with the results I obtained now? So what we started an experiment this year, trying to collect soil samples over the year, and we want to look at population. We try to look at certain processes also, because it's one of the critiques you can have on this work. It's very descriptive. What is this all about? What are you really doing? What is the importance of all this? And I would say we try to start with getting the techniques right, and then we can start to focus on some questions we have. So I'll mention some questions at the end of my talk. But we want to focus on nitrification and functionality. Extracting RNA from a soil is very difficult. If you look at literature, you will find some papers on it, but very little. Problem is soil conditions. And we are very lucky that Obregon, we succeed in extracting RNA. In El Batan, forget about it. So you check the literature. It's the same I mentioned bacteria, because extracting or amplifying DNA from fungi and microscopic eukaryota is not so easy. Archaea, we can do it, but it's a borderline. So look at the literature. You will see some papers, but if you try to compare, because we published a paper on fungi in Sonora and microscopic eukaryota, there's nothing to compare it really. Fungi, it's okay, you have some, but microscopic eukaryota, very little. Okay. If we have this question about copiotrophs and non-copiotrophs, then My idea was we get organic material. If we get organic material available, we'll get some copiotrophes. A rhizosphere is normally considered in organic carbon. So what we are trying to do now, how are these two behaving in the rhizosphere? There's a lot of organic carbon. Is correct? Environments. And also, if we apply organic material, a crop residue to the soil, who's going to degrade that organic material? Okay? This is fairly recent, and I haven't gone into all the details, but we measured population, vegetative flowering, grain filling. So the bulk soil rhizosphere, this dynamics a bit, and then again this another rather interesting development. I always used to work with SAS, and people convinced me that R is much better. It is a magnificent tool, but it takes some time first to adjust to it, because SAS is very straightforward. R is a bit strange if you're not used to it. Another problem I see with R, I can do this. These graphs are, well, the results were obtained in RI and then put it in Excel because I don't like the graphs. But you have about 6 or 7 packages that can do this. And I get lost sometimes. SAS is easy, it's straightforward, always the same. But R, the problem with SAS is all these biological techniques, they don't exist. And in R you've got all these updates, well, daily, I think. So my advice is people really want to work in bioinformatics, go to R or Python. I don't know Python because they're trying to convince me also to start working in Python. I don't know, presumably I will end up doing it, but anyway. But we get some clear changes again in population in the rhizosphere. So, and are they holds more or less? I mentioned that techniques there in R. Not too sure if it will all give better results, but and this is also very recent. Although I think the program or this was they started to write it when we were in Oak Ridge. They mentioned it. This is where you got your bacteria identified with ribosomal DNA. Sorry. And then you try to link these populations to functions. And that is very interesting because otherwise you've got 600 or 1,000 species. Bloody hell, what are they doing? Okay, I know Bacillus and I know Pseudomonas and I know Echis and I know that. But this becomes interesting. Now, I think they're developing it because RNA is not so easy to extract, but I hope we can link this with RNA. This is heat. It's just done, so I haven't gone through it, and these are some of the graphs in there where you can, I think, compare it. But I said it's just, I did it 2 days ago. And then this is another experiment we did with, these are all incubations over time. So if you see these 7 and 14, normally we have about 260, 250 data points. And then you see here we added bean material, and this is unamended, and then you see that the population, there's a shift in population. The story is I apply organic material and there's a shift in my population. Okay? As I said, we can confirm what we found in conventional tillage versus conservation agriculture. But as always, once you get some answers to your question, there's some other questions arising. And that is, as I said, I think the paper of Feeder 2007, where they defined certain groups as being oligotrophs and certain groups being copiotrophs. And then you compare Actinobacteria, this group, what is it, Micrococcacea, and then you got here Galenacea, and then you see these are favored by organic material you applied, and the others not. So the idea, yeah, it's not working. So I think it would be rather difficult to say that one bacterial group has these characteristics. I have to mention, when we go to a species in bacteria, then we talk about 97% of similitude. Yeah. We differ from chimpanzees, I think, 0.3%. So what are we talking about, really? Okay, but we are aware of this problem, but we should sometimes not forget it. Okay. As I mentioned, these groups are very easy to isolate, so they're well described. But in a soil, they're normally not that important, only 3% of them. And then we got the other bacillus of 5%, so what is all the rest doing? One of the other problems, when we write these papers, then normally we mention about 15 groups at the maximum. And normally you found between 500 and 1,000 genus. So what all the rest is doing, we don't know, and we don't even want to talk about it, because we would get a paper of I don't know how many pages. But anyway, so would you, the question was, would soil characteristics affect our behavior of certain groups when we apply organic material? So we apply organic material in one system, which is conventional beds, we add it in permanent beds. So we would hope that this group, which is Acidobacteria II. This is a very important group, but we don't know actually what it is doing. And then you see in permanent beds, when you apply bean material, their relative abundance increases. When you apply it to conventional bed, so there is an effect of soil characteristics. So we wanted to have some soil a bit more diverse to see how different bacterial groups respond to. And this, we used Chinampa, and we used Texcoco. And then this is quite nice. So you get a complete separation of your different groups, different environments. Yeah, one of the problems I have is with canonical correlations. When I do a principal component analysis, it's very nice. I go to canonical, and it's not so nice. So my explanation is there's a factor defining this that I can't, well, I don't measure it. Or is it soil characteristics I'm not aware of that is important, or it's interaction between my populations in the given environment. But I said I would mention the problems I see, but okay, I think we can, and this is then in the different ecosystems. Now, you have the Chinampa, you have the Batan, Texcoco, and then you apply material. How are these groups responding to material? And then you see Bacillus, it's increasing. Why? It consumes the organic material, that's very well known. But then you start to see these groups, like, I think I missed that one, which is a rather well-known group. And normally it is a copiotroph. However, in this system, in Texcoco, when I apply organic material, it participates in the degradation of the organic material. And when I look at El Batan, not at all. So, conclusion, these groups adapt themselves, or they have the metabolic capacity, but the environment, the competition with other groups defines how they respond. Yeah? So we made a little flow diagram of which organisms were favored along a gradient. So we apply here some organic material, and we measured populations day 1, day 3, day 7, 14, 28, 56. And then you see the groups that are favored, their relative abundance increase. This is Alomonas. And if we go to Batanda, we see the normal Bacillus. And some Streptomyces also along the line. Yeah. But you see there are different groups that participate or are favored by degradation. Okay. I think I remember, if I remember correctly, Titia said that bacteria in soil normally keep the genetic capacity. They don't shed so much of the capacity as you will see in the reactor or in my stomach. You know, there's a quick interchange of genes in organisms. So this is again 3 systems. I apply E material, and you get a shift in your population. Okay, as I mentioned, this is all very well, very nice. You've got beautiful BCRs, and you say, oh, we've got a shift. But it's rather descriptive. So can we learn for all this? First of all, I think we can use the techniques to answer specific questions. And one of the specific questions we are trying to address now is an obregon. It's a problem with dry sowing and wet sowing. Where there is an inkling that it might be a biological problem. That germination, plant emerges and plant stand decreased under dry sowing, but not in wet conditions. So we would treat them with pesticides or herbicides or whatever, insecticides, fungicides then. So we assume there's a problem there with. So I think with all these techniques we now have, we try to look if it's really a biological problem we're facing. Okay? Well, that's not that difficult. But this is not that easy. Due to soil conditions. There are some people who extract to metagenomics, or where you pay a lot and you extract all the DNA and then you analyze everything. Yeah, it's rather costs a bit. But then you need, and I'll mention that later on, then you need computer power and you need knowledge to all these pieces of 300 or 100 DNA pairs to link them. That's not that easy, neither. But anyway, we're working on it. As I mentioned, and then another study we started is to look at nitrifier populations. Why do we want to look at nitrifier populations? Because they're related to trace gas emission, greenhouse gas emission, nitrous oxide. Why do we focus on nitrifiers and not on denitrification? There are not that many. So we try to focus on something that is rather easy to study. Additionally, the genes or the proteins involved in the process are rather limited. However, for instance, AMOA, which is ammonium monooxygenase, where I thought there was one, now there are already 3 different types. So if you want to look at these things, these processes, you have to consider all the possible genes. So we're looking at permanent beds, conventional, burned, and then we have minus and plus, and we want to measure this. This is a first a preliminary experiment. We applied ammonium to these different sites, and what we see when fertilizer is applied in the field, then you see that a lot of oxidation of ammonium, so you have a rather vivid or active nitrifier population, which is not that. Yeah. Okay, we want to measure, or we measure that, we want to measure these gases, we want to measure biomass, we go for microbial structure, nitrifiers. This is not that complicated. As I mentioned, you already have 3 of them. I don't know how many I have of these. But then we want to follow up RNA. As I said, it's not that easy because it's much more fragile. You need to extract your RNA, then you have to convert it to cDNA. And you're really interested in the genes, or, well, the expression of genes that are involved in the process. And if you consider 95% of the activity is normal activity of the organism, it's not related to the process you're studying, you've got a bit of a needle in the haystack, I think. So, metagenome and transcriptome. It's not so evident, so we're trying to work on that. One of the things I haven't mentioned yet, but I think with all the changes in techniques we have, well, I mentioned it, we started 150 clones, then we went to 100,000, and now I don't know how many, 200 million. You need a bit of computer power. And we're only starting with that. So we'll start now, since yesterday, I think. Finally, after 2 years of negotiating, we can have a new option within the department, which will focus on bioinformatics, statistics, and supercomputing. One of the problems I see in statistics, personally, is time series. I mean, if you have a change over time, how do you compare these populations? What we normally do, what Richard Webster always told me, you just take the mean and you compare the mean, and that will be enough, and forget about all the rest. But I think we need some changes there. Supercomputers simply to get your metagenome together. Bioinformatics, there's a lot of dispute about the systems. You've got your sequence, and then you have different systems to verify or check or identify the organisms it belongs to. And so you have certain journals that say we want that system. Europeans normally go for the European system, and Americans the American system, and yeah, okay. But anyway, that's another of the questions we have. And then another thing we will try to do is we did a lot of these experiments with organic material copiotrophs, oligotrophs. We want to get it all together. It's about 20 experiments. We want to compare and see if with all these data we have, some bit of data mining, see if we can get something out of it. Okay, that's where we are at the moment. I must admit, as you can see, this is not my work. It's work of a lot of people. And I haven't mentioned them all, but would like to thank them. One of the advantages I think we have in Mexico, we have a lot of people. That's for sure. A lot of very enthusiastic people, very capable people. Funding is not always that easy, but okay, we'll get along. And we have no access always to the most sophisticated equipment. But okay, these sequences, we send them out to Microgen, Korea. I don't know if you read about the German group who now extracts DNA from soil, from our ancestors. And have you read about that? That now they can extract DNA from soil, because all DNA doesn't degrade in soil, so it is preserved. So if you look at Neanderthals where they walked, we can find traces of this DNA. Only mention it. The amount of soil you need to do this is not 20 grams like we do in El Batan, but it's a huge amount. And what these Germans have, they developed a robot. So you can assess or process an enormous amount of. But we have the people, they have the robot. We hope in 20 years' time the robot will be cheap, but all the rest will be rather expensive. But anyway, I would be very indebted for the field experiment. I think what Bram mentioned is true. It's a unique experiment. When I answer these questions, I have 2 replicates. I always say it's the only field experiment in the tropics that I have access to. Okay, there are 2 replicates, but well-maintained experiments for more than 25 years, correct, Bram? You don't find them. So if you want to study these things, I mean, okay, it might be useless, but I'm not so sure, then you need something you can rely on that's well done. Because if you go into the field and we compare whatever, El Batan, we compare forests, the variation is huge, enormous. And often you don't know what they did. So I'm grateful for it. And also when the money was short, they were more than willing to help me. I'm indebted to Silver for the infrastructure. And of course, CONACYT was very generous with funding us. I think that's it. Yep.
Thanks, Luc, and even that I was involved in the work, I still got some new ideas by seeing it presented like that. So, as always, you keep us on our toes. Any questions from the audience? Please use the microphones because we have over, I don't know how many people, but several connected via the webinar. So if you can use the microphone. Ivan.
Hi, Luke.
Hi.
Very interesting. I think I might have missed, when you collect your soil samples to do your extractions, what depth or from what part of the soil are those soil samples?
We normally work on the topsoil layer. Why we do that is because we hope we have the biggest diversity. I know there are papers published that you have to be careful with the profile. I'm well aware of that. But it all has to do with where do you want to focus on. If I said 200 samples to analyze, if I would go into profiles, then I get lost. It's always trying to,
So that's 0 to 20?
0 to 15, 0 to 10, 0 to 20. It depends a bit, yeah. But normally it's 0 to 10.
And then I don't know if you could go back to the soil tests of the 3 different locations.
Yeah.
I don't know if I saw Texcoco with a pH of 9 something, or maybe it wasn't pH, it went too fast.
Is it too acidic?
Too alkaline.
But I think you have to, Texcoco Lake, maybe give some more details on.
Yeah, Texcoco, the pH of 9 is not here at the station, right?
What, this?
No, the one at the bottom.
Oh, no. Oh, sorry. Oh, yes, okay. I have to explain this. Because you have these differences in El Batan. We see a difference. And so we see an effect of what we do on our population. So to stress, to go in an excess of variation, we went to soils in the area. It's a Chinampa. It's in Xochimilco.
Okay.
Texcoco, you got 9.4, which is not a lot, because in Texcoco we have 10.5, but we wanted to not to be too extreme. You know what I mean?
It's the Texcoco Lake, right?
Yeah, it's a Texcoco Lake.
Okay.
It's a lake. Yeah. And you have a huge salinity also. We did a lot of studies on Texcoco. That's why I included it.
So El Batan is I think if I'm right, El Batan is the CA plot from the long-term trial. Chinampa is Xochimilco, if I'm saying it right. And then Texcoco is the Texcoco Lake.
Sorry. Say sorry.
And one last question.
No.
Please.
What's your view on these biofertilizers that are dealing with these bacteria and they're being inoculated in the seed, etc.? Right?
Yeah, well, this is not well, it's personal, but I'm not the only one who would say that. I remember David Jenkinson saying that, and I remember Martin Alexander saying that, and I agree completely. I think it would be very difficult. There are 2 reasons. First of all, you have an equilibrium. You have a system, an equilibrium. You apply something, organisms. It's food. Second, if you have a soil, you apply it. How are you going to apply it where you need it? I can imagine you fertilize a seed, and then you hope. We did some experiments. We did an experiment with bacillus, and it was fantastic. It was the best. We had an effect for 1 month on the roots, and that was it. Now I'm actually doing an experiment with maize, with one of the farmers who has this brilliant system of biofertilizer, and we sterilized his consortium. And in the beginning, the sterilized consortium gave better results than the consortium. There's another reason why I'm very skeptical. For this experiment, we checked the literature. And it tells you quite a lot. Where was it published? Which journals was it published? I get very doubtful. I remember in Leuven, Bram knows Rule, but it was Karol Vlasak with Azospirillum in Brazil, and they had a positive result, so they published it, and they could never repeat it. The experiment we're doing is we using these techniques we have now to look more at survival. So we have this consortium, we'll characterize it, we'll see what's happening. Problem is always, okay, I'm working in greenhouse, because if I do it in the field, then again, you know the problems in the field. people do things you don't know. One of the other, that's the reason why I prefer to work with CIMIT. We started to work with somebody in, where is it? In road to Querétaro. I don't remember the name, Tepeji, nearby Tepeji, I don't remember the village. And you try to collaborate, and then they call you and they say, oh, we fertilized. Yeah, do you have sample? I don't have it. So you can't, and that's also one of the questions of reviews. Why did you do it in a greenhouse? Yeah, I can't say I can't work in the field because they don't allow me. It's not like Europe. I remember we measured some trace gases in Chiapas, in South Chiapas, and we left our PVC tubes in the field. They stole them. And we were communicating constantly with local people. It's one of the reasons also why I think field experiments and CIMIT are brilliant, and that we do some lab experiments. But it's very difficult to justify to a review who says, yeah. Okay, yeah.
Thank you for the talk. Just a comment, we actually also have a field trial that's been going on for 20 years in San Luis Potosí at the INIFAP station, so it might be interesting also to do some investigation there. They also evaluate conservation agriculture, minimal tillage, and conventional system, and 2 replications.
Okay. What kind of soil is it? So it's if we can extract the RNA, then I come immediately because it's rather no. It's not even. No, sorry. It's not even to do with the type of soil. It is at random. We had in the beginning, for instance, in El Batan, we sampled or you try to extract DNA. You got 9 replicate. Well, you got treatments. And all of a sudden, one soil, you can't extract it. Whatever you do, now we optimize that and blah, blah, blah. But if you go to conferences or congresses, there was one at Braunschweig last year in December, and then you hear the same questions. DNA not a problem, RNA, proteins, that people, huge amount of money, dedicated to extract proteins because that's the final solution. I mean, DNA, RNA, and proteins, and they got money. Europe, it's 1 million euros, but you know what I mean? These techniques are not yet what they should be, but I think they're brilliant. I love it.
Thanks, Simon, for your comment, and I actually think that several of the platforms that took some time to get them ready slowly are going to be ready to be put in these kind of networks, and taking advantage also of thinking all the PhD students that actually have been connected to the work, and that I think that's something we have to reactivate also in the team to see how can there be a PhD student working on San Luis Potosí, or so definitely Nele is working on that. Ivan is working on attracting more students. So I think that's how we can keep this kind of work going. Any more questions? Question over there. Somebody else? No? Somebody online? Sorry. Somebody online has a question? Now that I was recently following a seminar online, I know how frustrated it is. You want to say something and nobody remembers that you're actually hanging on your computer. No? One time, 2 times, last time, no question. Luke, you had a question.
If you have a specific question in your field experiment, I'm more than willing to investigate it. But the descriptive, I think, no, because we've done it. I think we should start use these techniques to really, I have a problem here. Can I answer it with my molecular biology? I think so. So if you have a question, more than willing. Yep.
Okay, thank you very much, Luke. And I think this is a clear example of research for impact through partnerships. When I'm asked what is the value of the long-term trials, I just show Luke's first slide with I don't know, I don't even remember, more than 30 joint publications and H-index of 14, and that usually helps to convince the more academically oriented donors. So, Luke, thank you very much. This has been a brilliant partnership, and there's more to come in the future.
Thanks.